Good morning, everyone. So thank you very much to the organizers for inviting me to present. Really been a great meeting so far, great educational experience for me. I'm a neurologist. I've been doing drug development for about a little over 20 years, but it's a new area for me, and been at GRIN Therapeutics in our body for about a year now, and it's been just a great, really a wonderful experience meeting the community and continuing to learn and hopefully working to develop a new treatment. So I'm going to talk to you a little bit about a drug called Radiprodil and the trial that we're currently doing. This is the outline of the presentation. First of all, I'll tell you a little bit about GRIN Therapeutics very briefly. And then I thought that it might be helpful to walk through how a drug or a compound, an idea really, becomes a medicine. Because part of that process obviously involves the community, industry, academics, advocacy, but also involves partners in government and regulatory authorities. And that part is pretty much many times a black box. And I want to sort of walk through a little bit of what that looks like and how that's approached. Then I'm going to talk about the NMDA receptor and GRIN-related disorders and why we're trying Radiprodil, and give you an update on the status of the trials ongoing right now. So GRIN Therapeutics, we're a New York City-based biotech company. It's overseen by Nervati Neurosciences, which is the parent company funded by a single investor, Blackstone Life Sciences. We're developing Radiprodil for gain-of-function variant, targeting gain-of-function variants. We'll spend some time on that, talk about our trial. The company was formed in 2019, or 2021, really, inspired by the 2019 GRIN conference in Emory. And it was the partnership that Nervati, with the formation of the leadership team in 2021, that really was the kickoff of the Nervati and GRIN we see today. And from the beginning, we've been coming to these meetings, and from the earliest stages, bringing the advocacy community in so that we can make sure that we're doing the right thing by the community in our studies. So now I'm going to walk through a little bit about the traditional steps in development of medicine and just talk about the regulatory process. This is at a high level, what it looks like, and it looks very linear. It never is this linear, but this is sort of the concept where you have the idea, the basic research, the sort of the selection of what you're trying to do, and the drug discovery process where the biological problem, the disease is, the better the biology is understood, chemicals, proteins, genetic medicines are all designed at that early stage to try and address that biological problem. And that could take a long time, depending on how much is known about the drug. But once you get through that part, you get into this stage called preclinical development, which is also people call toxicology. And that part, you have what we call pharmacology characterization, which means you do studies to figure out how the compound does in animals. How do animals process it? How does it act? What does it bind to? What are the types of things that it might do in humans? And actually that gets to what dose levels are likely to be active in humans. And that requires, again, understanding how in a animal, and usually rodents and larger animals, tolerate the molecule. Then you go into toxicity studies. So this is the early signs, looking for what are the risks that this compound may pose to humans. And that ends up being single dose studies, again, usually in 2 animal species, a rodent and then in a larger animal like a dog or a non-human primate. And these are looking at single doses and multiple doses at possible risks, and also the highest doses that are possible in animals before you start seeing adverse effects. Because the idea here is to try and create a window within which you can safely dose in human beings, and that really is understanding how high you can go. And then you want to try and mimic human dosing as much as possible in terms of the levels and how often it's given, to, again, try and predict risks so then risks can be mitigated when you get to humans. once you have all of that done and you have that preclinical work, in addition, have work on how to manufacture the drug, because you want to be able to have it done in a high quality way that doesn't pose risks to humans, you file a clinical trial application, or you set up a clinical trial application. That's an IND in the US or a CTA outside of the US. And that really is a collection of everything you have to date in terms of the justification for why it should be given to humans with all of this preclinical toxicology and pharmacology work I just described. Then you move into the first in human testing to try and confirm some of the biological effects you've seen in animals. And that is, again, focused on safety and pharmacokinetics, which is another word for how the drug behaves and acts within the body. That can be done as a 1A study, which is really your traditional single ascending doses, 1B, which is multiple ascending doses. And these are general terms, but there's a lot of variation within there, the types of things you can do at those different stages, especially in the setting of a rare disease where the number of patients available for studies is very, very small. So there are ways that this can be somewhat modified. After you understand how it behaves in humans in a single and multi-dose, and that can sometimes be healthy volunteers, that could be patients. Now you're trying to understand how it behaves generally in the disease of interest, in patients with the disease you're targeting, because maybe it'll behave somewhat differently. And then you could do single doses there or multiple doses in an early dose finding, and that's called phase 2A. But the whole concept of phase 2 is what's called proof of concept. You wanted your hypothesis that you've been developing since the early days of animal testing is now for the first time coming into humans in a way that you could try to evaluate whether all of that hypothesis, all those ideas translate. And that's usually in a repeat dose study. They can be anywhere from 3 to 6 months, typically has larger number of patients. You may, because animals are not humans, you may have to do more doses, a dose range to try and understand about whether your modeling and your ideas from animals translates. And then you want to, basically the goal is to try to figure out, before you now dose a larger number of people in a phase 3 study, which we'll get to, you have a good idea of the dose levels and your safety profile, because the goal is to limit the number of doses and frequencies that you're exploring in your final study to the doses and frequencies that you hope will someday translate into how you treat people once the drug is on the market. Now, especially in the rare disease area, there's all sorts of combinations of this. Often phase 1 and phase 2 are combined, and that's what we're doing in our study, and that's quite common, where you get a phase 1B, 2A, which is a multiple dose, single dose, and a dose ranging finding in patients, going directly to patients, and again, looking for signs of efficacy. And then you have phase 2, 3 sometimes combined. There are a variety of ways that this can be done depending on the disease area, and also depending on whether you have enough safety and tolerability data, or you've mitigated risks enough to skip or combine some of these steps, because the goal is to do this at a pace and in a way that is safe for the patients in the study. It cannot be done in too fast, because that's usually where things go wrong. And when something goes wrong in this phase of development, it could set back the development for years or forever. It could be the end of a program. So we try to do this in a way that's methodical and driven by the data. So then what happens if we're all fortunate and our clinical studies show that there is efficacy and safety, so a favorable benefit risk for a compound? That's where you go to commercialization, which is essentially the submission and the filing for approval. And in the US, that's called an NDA, or a new drug application. A standard review is about 12 months, a priority review, which is an accelerated course for a disease where there's high unmet need or no available treatments. That typically is about an 8-month clock, with the FDA reserving the right to extend that by at least 3 months in the setting of additional data needed or time for manufacturing, something of that nature. So they can extend that if necessary. And in the EU, that's called a market authorization application, an MAA, typically 12 to 14 months. Same concept. That's a little bit more driven by how many questions there are and how quickly the companies are able to respond to those questions. So that is the process. Now, what it looks like is often this is what's called the CTD triangle, or the Common Technical Document Triangle. This is sort of the structure of a submission, where all these things I was talking about in the preclinical stuff, you can see are the foundation. That's the bottom of the triangle, where you have your toxicology studies, your pharmacology studies, some of your clinical studies, all the details. The quality, again, that's your manufacturing information. Then this is all summarized for the regulators in that sort of middle section, which is called Module 2. And then luckily for all of us, the regulatory authorities around the world through this ICH, or Conference for Harmonization, International Conference for Harmonization, standardize how this works. So once you put this together, the regulatory authorities globally have agreed to accept it in this structure with these components. And Module 1 at the top of the tip there, that's the part that every country gets to determine what is specific for them. So every country reserves the right to ask for additional information, but the foundational stuff that the application is based upon is the same for all regions. Now, in terms of the scope of this, I mean, the last one of these I did, if people count in pages anymore, was 4 million pages. So these are big documents. They take this, and the fact that it takes 8 to 12 months to review it is actually quite remarkable. But that is what goes into an application, and you can imagine that does take time. So, and it takes a lot of work. So this is sort of what goes into it. Along the way, there are interactions with regulatory authorities who are giving you feedback. And I would encourage, and I always do when I give this presentation or variation to whatever therapeutic area, engagement of the regulatory authorities is essential throughout this. Especially in the setting of a rare disease, because they don't know it. They can't know. Remember, they're reviewing everything on every disease in a certain area. Everything in neurology, it's everything from stroke to the ultra rare to the new. So the more the community can educate and engage along the way, you'll find that that's extremely helpful when we get to this stage, because the importance and the unmet need in an area is often unknown to the authorities. And so I always encourage people to engage, and often the regulators will attend conferences like this, or have meetings with the community to help them understand, because they want to understand, because they want to do the right thing. So that's always a plug I always give in regards to the other stakeholder in this process, which again is the regulatory authority. So that's what I have on the process, and we can certainly come back to that during the questions. Now I'm going to focus on NMDA receptor and Radiprodil. Now, I'm lucky to go at the end of the meeting, day 2, so I don't have to spend too much time, because by now I think everybody sort of knows disease and the receptor. But just as a quick review, here's the receptor. This is the NMDA receptor that we all know is encoded by GRIN genes. And this shows the NR2 subunit there with the glutamate binding site and the NR1. And the target this is the target of where we are targeting Radiprodil, and we'll come back to that. And in our case, we're talking about gain of function, but this is a diagram that we like to use to explain the difference between gain and loss of function. And if you think about essentially a barnyard filled with looks like hens and chickens, or animals of some form. When you think about a loss of function, you can imagine the gate somewhat being stuck, and the flow that's natural between the barnyard and the field outside is constrained, versus when there's a gain of function, the latch is open in the wind and the flow is unregulated. And that's sort of, as we think about how to understand this process, the idea here would be in each case to target the issue at hand. And in setting of gain of function is to try and restore that regulation so that this can function, the gate can function in the appropriate way, and the way it should be, again, to regulate that natural flow, in this case through a channel. So when the NMDA receptor is overactive, that is part of the problem, as you're all aware, in GRIN-related disorders. The cells of the brain who naturally communicate through this highly regulated process have trouble communicating. This leads to problems developing. And that leads to the presentation of developmental and epileptic encephalopathy, as we all know. I don't have to describe this disease to this community, but I think it's fair to say that this combination of seizures and behavioral symptoms are the result of this dysregulation of the receptor and significantly impactful. So there are no therapies specifically to treat GRIN-related disorders, and as you all know, quite resistant. The disorders are often quite resistant to standard treatment, and then that's where our approach of targeting the receptor itself in a selective way is our hope that will lead to this restoration of regulation. And that's what's shown here. Radiprodil target, it's a negative allosteric modulator, so it binds to the receptor and helps reduce overactivity. It targets the NR2B part of the receptor, as shown here in this diagram. And our hypothesis is that when you selectively target NR2B and you have this overactivity as a result of the mutation in that area, that you can reduce the flow, you can regulate the receptor again, leading to fewer seizures and improved development and behavior. This is the study on a high-level overview of the study we have ongoing. Again, a phase 1B, which means it's single ascending, also going into multi-dose to look at the potential biological effects. This is currently enrolling in Europe, with other countries gradually coming on board. The primary objectives, consistent with the stage of development, safety and tolerability, optimization of dose range for future studies, but also we're measuring the impact on seizures. We're looking at EEG, behavioral symptoms, sleep, motor function, as well as caregiver burden and the impression of effects. The inclusion-exclusion criteria, as I mentioned, gain of function, and we're targeting patients between 6 months and 12 years, in one of two cohorts, a cohort with at least one seizure per week, and another cohort where significant behavioral symptoms that may not necessarily meet the seizure threshold. I just posted on clinicaltrials.gov the numbers there, and there's a lot more information there and in other sources. This is a high-level shot of where the study is ongoing right now. The centers are here with the green centers active and open for screening. An important part of this study is that gain of function is an essential requirement. And so the way this is being done is that even if a parent has been told at one stage that their child has a gain of function variant, we just have to verify that centrally through a committee that's been a panel of experts that has been assembled based on a recent publication by Myers and all. They'll apply that assessment and will communicate back to your physician whether the committee agrees that for the purposes of the study, it meets the gain of function definition, because we are using a pretty tight definition simply because for an early phase study, that's not an area that we want to be flexible on. I mean, again, it goes to that area of making sure that this stage of disease, we are very careful about the right patients coming to the study so we can better understand whether there's potential benefit, what the risks are, and then plan future studies. So, I mean, this is a picture that's taken at the GRIN 2B meeting in the US. I think it's 2021, I believe. And many of you were there. And we often get the question, what can I do? How to get involved? Obviously, you were all involved, so I don't have to really tell you as much as others, but really staying connected and understanding what's going on and asking your questions and trying to influence how things are done, staying aware of developments is really a critical aspect of this and is going to help us move things forward for you. There's a lot changing. I mean, I've just seen in the past year that I've been involved in this community, things are changing rapidly. There's a lot of developments to keep up on. Just stay alert to that, read about the trials, our trial and others. There's developments all the time. We have, for our study in particular, we have a screening tool available at that website, thehoneycombstudy.com. And you can always reach out to us through patient advocacy. This goes to Hilary Savoy at Nervati and GRIN. And, we're there to answer your questions. And, we are there to ensure that you get the best information about what's going on in our study, but also in the community at large, and at least, and of course, we're always available to answer questions on it, pretty much anything. So, this takes a lot of work to get to this stage in the study, and it involves a lot of people that I want to thank, that even at this stage where we don't have data, the amount of work, the amount of time, the amount of contributions, again, from this community into our study is much appreciated. So thank you for those who have contributed, and we look forward to working with you to move this forward. That's it. Thank you so much.